So according to my computer, it says that it's 9 o'clock, and are the loudspeakers actually working now on my mic? Yes? No? No. Does it matter? You can probably hear me anyway. Hopefully, it looks like at least 53 of you have checked out the lecture from last time, which is on YouTube. I'm going to try and keep posting all of them. There was an issue with the resolution on there that is mea maxima culpa. I'm not still quite sure how to get all of this recording, video editing software to work. Should be much better today. The other thing I'm going to try and do today, and this is always an issue when you're trying to do screen capture, is to use a much bigger cursor, which you can actually see, as opposed to laser pointers all the time. So I'll try and do that as things move along. Have people had any issues with recording, other than the resolution issue, which I'm working on? No? Seems to be okay? Yeah, in the back. I try to, yes. Please remind me if I don't. So, like, yell, jump up and down, wave your hand, something like that. Yeah, so the question was, or the comment, please get me to try and repeat all the questions, so that way it actually ends up on the recording. Okay, other things. So, everyone have their clickers out? Yes? Happy? Good? No? Let's see if this is actually going to work. Yes, it does. So, which of the following organisms is the smallest eukaryotic model genome? Escherichia coli, bacteriophage T4, Saccharomyces cerevisiae, Centrobritis elegans, or Mus musculus. And I could actually start that. That might help, so you can actually do your countings here. And again, now up here is the countdown. I'm going to give you a minute and 30 seconds. And again, feel free to talk to your neighbors. And you can change your votes any time until that time is actually up. And you can always guess. These don't count yet until next week. Okay, now we're done. Let's see if I can actually show these. Yay. Okay, most people seem to think and agree with me that Saccharomyces cerevisiae has the smallest genome of eukaryotic model organisms. That's true. I just wanted to mention here, how many of you made it to the seminar yesterday, the biology department seminar? I was there. Talked about the model organism E. coli to understand some really fundamental aspects of DNA repair. And so the idea there is a lot of the stuff, again, we're going to be talking about in this class, including DNA repair. How do you stop everything that's going on in terms of the cell to turn on repair processes or not turn on repair processes at the wrong time? You can learn a lot by studying E. coli. And so that's part of the idea of model organisms. This is not just some crazy thing we bring up in class so that you actually have to try and figure them out. So next question. Start again. Interactions between biological macromolecules are best described by hydrophobic interactions, hydrophilic interactions, amphipathic interactions, molecular complementarity, or dissolution. Okay, what do people think? Seems to be pretty evenly divided between amphipathic interactions and molecular complementarity. Amphipathic interactions is great for interactions with water, but there are certainly interactions between biological macromolecules that have nothing to do with water. Which are those? At least 53 of you looked at the lecture last time, right? So what kinds of interactions between biological macromolecules don't depend on water, which is, amphipathic and hydrophilic, hydrophobic? So enzymatic function was a suggestion. I would say van der Waals interactions. Those have nothing to do with water. It's just atoms coming close to each other, very close to each other. And so really molecular complementarity is a better answer for this question. So let's see if I can get this to work. Yay, good, it did. So now we can move along and talk a little bit more about some of the chemistry I think you need to know for a molecular biology course, and then move on into some of the proteins and the mechanisms by how they really get put together. Any questions about sort of model organisms, bonds, chemical cellular components? Water, of course, is really where it's all at. A lot of these clicker questions are usually going to be very closely based on these. We really didn't talk much about energetics. We'll talk more about that today. So are there any questions on last lecture before we move on? Okay, so today we'll talk about chemical reactions at a very basic level. Again, this is what I expect you to know for this class. For chemistry, certainly there'll be a lot more involved. And then they'll talk a little bit about kinetics and how fast reactions take place. In biological systems, that's almost always going to have to do with catalysis. That catalysis almost always happens with proteins. And so we'll start talking about proteins and continue for the next 2 lectures next week talking about these proteins. Start out talking a little bit about protein structure, particularly in terms of secondary structure. And there's always seems to be a bit of a confusion between what's a motif, what's a domain, how all of these things fit into individual proteins. So we'll talk a lot more about that. Basic message there is a domain has a structure in and of itself, and a motif is a collection of secondary structures. If we get to it, and we'll see if we do, we'll talk about protein folding, basically how you go from your linear amino acid sequence to a tertiary structure, and of course that structure leads to function. By the end of the term, you'll all get that down. So first we'll talk a little bit about reactions. This is just chemical reactions, pretty straightforward. The key here is to really understand that there's a difference between rates and rate constants. And I think the textbook does a really quite good job. There are parts of the textbook that are really, really nice, and other parts of the textbook I don't like as well. I think it does a really nice job of discussing these aspects of chemical kinetics and chemical reactions. So here, if you look at the very beginnings of a reaction where you have reactants, all reactants, no products, then you look at the rate of reaction, and this is the rate of reaction. First, you're going to be all moving from reactants to products. So that movement of reactants to products is always going to be in the forward direction. But as soon as you start to make product, some of that is going to go backwards. All chemical reactions are reversible. And so eventually, you'll get to a certain point, and this is going to vary, of course, from reaction to reaction. You'll end up at chemical equilibrium, which is where you've got the rate of the forward reaction being equal to the rate of the reverse reaction. And so there's no change in the net reaction rate at this point. So that's basically all you need to know about chemical equilibrium. If you actually now use the math to look at this, you can get an equilibrium constant, which is dependent on the concentration of your products and your reactants. Here is just a very general case, 2 products, two reactants. Could have drawn this out. There you have a rate constant, which is the forward rate constant. That's going to be how much product you're actually making, and a reverse rate constant. You just take the ratio of these, the forward rate constant to the reverse rate constant, you end up with your equilibrium constant. That equilibrium constant is going to depend, of course, on concentration of products and concentration of your reactants. In cells, you're almost always not at chemical equilibrium. And a lot of people have said, if you're at equilibrium, you're dead from a biological point of view, because that's, say death is equilibrium. Some people talk about things that way. But very often you have concentrations of various different chemicals in cells, or even in an organism for that matter, which are kept at a pretty steady level, so they don't change very much. But that's not a reaction rate which has gotten to zero. It's just a concentration of one particular product which is at zero. And these are often intermediates in various different pathways. If you're interested in more about this, take biochemistry, and it talks a lot more about this. But just in a pictorial form, or semi-pictorial form, equilibrium is here. You've got our two rates. This is the top arrow is going to be your forward rate. The bottom arrow is going to be your reverse rate. At an equilibrium, again, where these rates are equal, no difference between the two, you're going to have a certain concentration of your reactants and a certain concentration of your product. So here we're at equilibrium, death as far as biology is concerned. So down at the bottom here, this is what's usually happening in a biological system, is you have multiple coupled reactions with each other. And so what that means is that B may be at a constant concentration inside the cell, but it's not at equilibrium, because they're just going to depend on the concentrations of A and the concentrations of C. And any of you who have seen the pretty famous now poster, and it could be about the size of this screen probably, with really small font sizes, all of the biochemical pathways that you have inside a cell. And each of the individual atoms and molecules that are involved in that. They're all linked to each other. So that's what's happening inside the cell. I want to talk a little bit more about a very specific case of equilibrium constants, and that has to do with binding. And the binding constants, very often people talk about the KD, or the dissociation constant. This is basically looking at interactions between two molecules. And yeah, that's really what molecular biology is all about. It's interactions between two different molecules. People very often will talk of these as a ligand relative to a protein. So here we've got L for your individual ligand, P for the protein. And dissociation constant is just concentration of the ligand times the concentration of the protein. These are now free, not bound to each other, divided by the concentration of the complex. Again, just like we had for equilibrium constants, that's the dissociation constant, the now it's the binding constant relative to the on binding constant. If you now have a concentration where you have half of your protein bound to your ligand, that's going to give you, L is equal to KD. Now, it took me a while going through the textbook to remind myself exactly how this is going on. Basically, if you think about, okay, LP, that's your complex, and P is going to be free protein. So half of your P is bound. That means that LP is going to be, excuse me, the same as your P. Half is bound, half is not bound. And so the bound is LP, and the unbound is P. So you end up with KD is equal to L. So your free ligand. And this is really important for thinking about how tightly you have interactions between different molecules, and usually in the case of molecular biology, this is going to be in macromolecules. So if you have a nanomolar KD, that means that half of your protein is bound when you have nanomolar concentrations of your ligand. And that means it's going to be really tight binding, because they have nanomolar concentrations of free ligand. There's very little of that free ligand around. It's almost all bound to the protein in this particular case. If you think about millimolar dissociation contents, those are relatively weak. So questions on this? This is kind of a bizarre concept, and I don't think it gets covered terribly well in other courses. So, and again, please feel free to send me emails, discussions, etc. We can take a look at it. Another aspect of equilibrium that's really important as far as molecular biology is concerned is the pH. Hopefully this is all review for everybody. It's just the negative log of the free hydrogen ions, really, of course, hydronium ions, as we talked about last time. Neutral pH here, pH 7, just for some comparisons. The interior of the lysosome, which the book pushes as being really low pH, that's actually 2 orders of magnitude higher pH than my favorite organisms, which like to live at pH 3. So there's some interesting aspects about that as well. But more importantly for this course really has to do with titrations. And you remember last time we talked about the acidic and the basic amino acids, which are present inside the cell, inside any given protein, and their ionization states. Well, if you look at the ionization states, it's much best understood, best understood for various different chemical solutions. Here we've got a carbonic acid. If you look at carbonic acid, this has a pKa, which is again just the association versus the dissociation of this free hydrogen, which we have here. If you look at concentrations, various different pHs here. The pKa is at 6.4. That's when you're half dissociated. So half of that is going to be in your product, half of it's going to be in reactants, and of course you can always flip these things around. They go back and forth. We've looked at this already for those acidic and basic side chains, but also in terms of the histidine, which has a pKa which is very close to 7. And so that can be charged or uncharged by just changing the local pH a very little bit. Now, of course, these pKa's association, dissociation of hydronium ions can be a big change in terms of all of the other molecules which you have inside the cell. So one of the really critical aspects of all biological systems is that you have these so-called buffers. And basically all that a buffer is it's something which takes your free hydrogens and instead of allowing them to interact with whatever other side chains, whatever other molecules you happen to have inside the cell, it basically associates those free hydrogens with that particular acid or base which happens to be there. And so all that this really does is it means that if there are some changes that happen inside the cell, it's not actually going to change the overall pH, which is those free hydronium ions, that much, because all those free H's and OH's are getting picked up by the buffer. And so one example here, this is acetic acid, where you have a pKa of 4.7, but basically between here, pH slightly below 4 and a slightly below 6, you're going to have not very much change in pH, even though your fraction of dissociated acetic acid has changed from 10% to 90%. So basically, it's sort of they call it a buffer. You can also think of it as a sponge, just sucking up any of those extra things which happen to be around in the cell. So any questions on sort of the equilibrium, pH, etc., before we move on here? So now I wanted to talk a little bit about energetics, having to do with how reactions take place. Again, hopefully this is all review for you. A energy yielding or spontaneous reaction has a so-called delta G, or free energy, which is negative if it's spontaneous. If, however, it's a non-spontaneous reaction, you have to put in energy, that delta G is going to be greater than 0. Almost all important reactions for biology, particularly molecular biology, and really thinking about making macromolecules, these are all going to be endergonic or delta G greater than 0 reactions. If you look at delta G, it's all about the heat or the enthalpy and entropy. Now, one of the main reasons that in most of biology these are now all going to be positive is that S is almost always negative. If you think about entropy, and again, people talk about, if we're at equilibrium, then entropy is really high. We're not alive. And so putting things together, organizing things, you're going to have negative entropy. And of course, negative entropy means you're going to have a positive term right here. So these delta G's are related to the equilibrium constant, which I think is just totally cool. But just if you look at the reactions here, the delta G overall is going to be dependent on your products and your reactants and a fundamental delta G0 for that one particular reaction. So at standard conditions, you're going to have just delta G0, and then any changes are going to be put into this component, R being the gas constant, temperature, and the natural log of the ratio of the two products. You can get delta G0 from your equilibrium constant or vice versa. So the equilibrium constant is this exponential reaction. Your delta G0 is this log reaction here relative to the equilibrium constant. So you can get energetics from your equilibrium constant and vice versa. So you only really have to measure one or the other. So they're directly related to each other. So why do we care so much about delta G's? Well, A, of course, these are going to be spontaneous or non-spontaneous reactions, but also almost all reactions have some kind of, it's called activation energy. So you have to get from your reactants to the products, and again, it's a very similar kind of graph here. Delta G, or the free energy on the y-axis, x-axis is going to be your progress of your reaction. Reactions to products here, this is moving in a negative direction. So this should be a spontaneous reaction, right? Except for the fact it doesn't necessarily always happen immediately like that, because you have some kind of higher energy state that this particular molecule needs to go through. And we talked about this already when we talked about electrophiles and the nucleophiles last time in lecture. And basically getting over, as people talk about, one of these high energy intermediates is usually very, very difficult and requires, again, a lot of energy input to do that. So in most biological systems, you don't have non-catalyzed reactions, and the catalyzed reaction just lowers the energy of this transition state. And now what does catalysis? Well, these are the enzymes in biological systems. Almost all enzymes are proteins. We'll talk about some RNA enzymes later on in the course. But basically, all of these, again, mostly protein enzymes, they will bind to one or very often two molecules and then stabilize some kind of transition state, help that reaction to go, and it's only helping the reaction to go. That reaction, there's no change in the overall delta G of reaction here, just lowering that transition state energy. And then you end up with a product. And we'll look at these in much more detail next week in terms of how all of these things work. In general, anytime you have an enzyme, it's got an ACE at the end. And for the most part, enzymes are involved in breaking things. So here, classic example on the right-hand side, we have a substrate, binds to an enzyme, that then gives you two products. So here, hydrolase, actually it's not breaking water, but nucleases, they break nucleic acid. Proteases break proteins. So phosphatases break off phosphates. ATPases break down ATP. So, again, ACEs gives a good idea of an enzyme, and most of the time what's happening there is that we have some degradation of one biological, and often a biological macromolecule. So why do we care about all of these things? Well, as I mentioned before, most biological reactions are energetically unfavorable. So why do we even have life in the first place, if all of these chemical reactions are unfavorable? Well, and that's because almost all chemical reactions are linked to each other. You've got multiple different chemical reactions. So if you look at an overall, what we like to call a pathway, where you go from one particular substrate to one particular product, overall, this is a energetically favorable pathway, and you can think about it as like eating food. You eat your food, and that goes from here, high energy, to low energy, and in the process, you can go to the gym, and so you can actually then use some of this. And so these individual biological reactions, for instance, A to B and C to D, are unfavorable reactions, but B to C and D to E are highly favorable reactions. And they have to be more favorable than these unfavorable reactions in terms of the energetics. And so you have to be able to somehow put these reactions together, and that's what we call coupled reactions. So the next couple of slides are going to talk about each of those processes. We already talked about the high energy, quote unquote, bonds that you have in ATP. They're really not that high energy relative to covalent bond formation. So we said before, most covalent bonds are 80 kcals per mole approximately, and ATP, that hydrolysis, gives you only about 14, again it depends on the conditions, exactly how much energy you get out of that reaction. So somehow you've got to couple a positive energetic reaction, i.e. with a negative delta G, to something which has a positive delta G. The way that's usually done in biological systems is the so-called precursor activation. We talked about small molecules last time. Very often it's the small molecules that then get incorporated into the larger molecules, and we'll look at that in more detail here in the next couple of slides. Another way of just looking in general at these coupled reactions, here we have a reaction of C going to D, which has a large negative delta G, and then the gear right here, that's the coupling between the two reactions, which now gives you a positive delta G and allows you to have this unfavorable reaction of X going to Y, because again you have this reaction which is giving you enough negative energy to make that coupling. Let's take a look at one example of this. Here we have glutamic acid, which is one of those amino acids which is a charged side chain. In this case, it's actually not being shown as charged. Here in that glutamic acid, you can activate that by putting on a phosphate group at the end here. And this phosphate group, the high energy intermediate now, allows you to undergo this otherwise really energetically unfavorable reaction. So what we've done is we've taken the OH group on the glutamic acid here, replaced it with an ammonia. You can't do this reaction directly, going from glutamic acid to glutamine, unless you now couple it to the hydrolysis of ATP. Now giving the high energy intermediate and then being broken down. Probably the best understood of these are the polymerization reactions, and we'll talk about these probably ad nauseum as far as the rest of the course goes along. Look at, in particular, proteins and nucleic acids. Here, for your protein, you have an individual polypeptide chain present down here at the bottom with OH on the carboxyl end. That's now going to interact with the amino end of an individual amino acid. You're going to lose water in this reaction. That loss of water is clearly you're going from two different things to one thing. Entropically, this is a really negative reaction. So you have to now couple nucleoside triphosphate hydrolysis to getting the polymerization taking place of these molecules. Same thing is true for nucleic acid. Here we have one piece of nucleic acid an individual nucleotide that then gets added to this molecule going from 2 to 1, and again, energy from nucleoside triphosphate degeneration. Same thing is true for polysaccharides as well. That's it for chemistry. Whew. Okay. Any other chemistry you need to know? Hopefully you know it already. Good, so let's move on and talk about proteins. Some of my favorite things. And really it's because proteins do most everything in molecular biology, with the exception, again, of some of the RNAs. But proteins do a pretty darn good job, and in fact, in most cases, proteins work much better than RNA in terms of how life functions, or at least life as we know it functions. So proteins are important for regulation. That's the very last part of the course. They're important for structure, and that's I talk about cellular structure, but also macromolecular structure. Collagen, for instance, is the most common protein that we have in us. Basically makes us not be a blob. Movement, moving things around inside of cells, outside of cells, all that's dependent on proteins. Catalysis, we just talked about, lowering the activation energy for various different chemical reactions. Transport, moving things in and out of cells, particularly those monomers, which you can move easily. Polymers are, of course, a lot harder to move around in cells. And signaling, which you can to some extent think of in terms of regulation. So proteins have all of these really interesting functions, and we couldn't imagine life really now without proteins. How do you make a protein? Well, it starts out with a string of individual amino acids which are put together. That's your primary structure. That then goes to secondary structure. We'll talk about those individual things in just a second. That will then lead to tertiary structure. And really, I'd say tertiary structure is really a three-dimensional structure, and that's usually a relatively stable state. In some cases, that tertiary structure gives you the function directly, but in a biological system, if you think about inside a cell, it's actually really packed in there. We always have these nice slides with one protein here and another protein here, all nicely separated from each other. That's not what's going on inside the cell. They're all really packed together really closely next to each other. And in many cases, you actually have to have these molecules right next to each other in order for them to function. And in some cases, we know that there's very specific interactions. Otherwise, you don't have any function at all. And those are the so-called quaternary interactions. And then many of those will assemble into these larger assemblies. So let's look at those individual cases right now. Start out again with primary structure. This is just the sequence of the individual amino acids. What they should have put on here is that this also, of course, has an N-terminal end and a C-terminal end. So each of the monomers has molecular biology polarity, and from one end to another. Those then form up into secondary structures. The most common of those are alpha helices and beta sheets, which we'll look at in just a second. These secondary structures come together to form first motifs, so there should be a little piece in between here. Multiple secondary structures coming together. Then you have tertiary structure, and in those tertiary structures you have domains. These are individual pieces of three-dimensional structure which have all come together. In some cases, but certainly not all, these individual proteins, and this is just one polypeptide chain. It's got an N-terminus here. Not quite. Here's the N-terminus right there, and a C-terminus right there. You can have multiple polypeptides coming together in quaternary structures. So if you think about a tertiary structure, that's going to be one polypeptide. It's not going to be multiple different pieces. So multiple different pieces coming together, that's going to be your quaternary structures. So look at that here again. Again, hopefully not beating too much on the dead horse here. We've got the carboxy terminal end, the amino end of two individual amino acids. These guys come together, form a peptide bond. This happens again and again and again. You end up with a polypeptide, which again has a amino end and a carboxy end. Really important for the actual function of the proteins. We mentioned there are 20 common different amino acids that you start out with up here. They're different relative to these R groups, the green blobs. that we have here. And those then are the differences that you have between any individual amino acid. But the backbone here, we've got your aminos and your carboxyl groups. Those are exactly the same. Each of those amino acids is going to be identical to each other. So you take this string of amino acids, your primary structure, and then you start to put it into 3 dimensions. And there are really sort of 2 main ways that you put these guys together in 3 dimensions. The first one is the so-called alpha helix, also discovered or at least first characterized by Portland's most famous scientific son, Linus Pauling, whereby you have the individual amino acids that interact with the amino acids which are present on that same piece and usually very close to each other, in this case about 3.5 of the amino acids apart relative to each other. These dotted blue lines represent the hydrogen bonds, and again it was Linus Pauling who first described the hydrogen bond. We've got hydrogen bonding between the hydrogen atom on the amino group and the double bonded oxygen that you have on the carboxyl group. This, of course, is backbone. All of the amino acids have these. So at least in theory, all amino acids can interact together this way. So it doesn't depend on your side chains at all. One of the really neat things about having a structure like this is what does it do with all of your side chains? If you look at where all of these green things are here on the outside, they're all sticking out. So all of your side chains are sticking out from the alpha helix. And this is really important because that's, of course, where your diversity is. That's where you can now interact with other things, interact with other molecules, other parts of the same molecule, etc. So you've got the backbone interacting with itself over relatively short distances, giving you the side chains which are sticking out on either side. The other main form of secondary structure is the so-called beta sheet, which is just literally multiple beta strands that come together, and they just called these alpha, beta, and gamma originally. Linus Pauling, when he was doing these things, early 1960s, if I remember correctly, looking at various different proteins and trying to then figure out how they're actually interacting with each other. Here you also have hydrogen bonds between the backbone, either amines or carboxylic acids, but instead of being relatively close in terms of your primary sequence, they're far apart. And they actually could be completely far apart, even in some cases on completely different molecules. And so here you're having interactions, and again, hydrogen bonding that forms between 2 pieces of your primary sequence. This can either be in a anti-parallel form here, going from the N-terminus to the C-terminus, N-terminus to the C-terminus, or you can also have the parallel form where you've got N-termini on both sides here. But just like the case that you have with the alpha helices, where you're sticking those side chains out, the alpha helix, you're sticking them out on the outside, just around your alpha helix. Here you're sticking those up and down. And again, allowing the interaction of these side chains with other macromolecules and other parts, potentially, of actually even the same macromolecule. Here's just that example of parallel versus anti-parallel. Here again, we've got our C-terminus at one end exactly the same on this side, and here varying from C to N-terminus on either side. Ends up giving you exactly the same thing. Again, side chains sticking out, backbone interactions interacting with each other. There's one more form of secondary structure that they talk about in the textbook. It's not really sort of considered as a standard secondary structure. The 2 main ones are alpha helices and beta sheets. But you also have these so-called beta turns. There are really 2 main flavors of beta turns, 2 different kinds of beta turns. There's all kinds of different loops, however, that you have between these individual secondary structures usually. These are some very specific ones. This particular one has a proline in the middle of it, and proline, so remember, it's one of those weird amino acids that I had down on the bottom of the slide before. That's because it doesn't have that free R group hanging out in space. It actually hooks back up to the backbone. And so it actually provides a lot more rigidity and constraint to the flexibility otherwise that you would have in a peptide bond. And we'll talk about those peptide bonds in just a second here. Here, proline automatically gives you a kink in that backbone. And then if you have some hydrogen bonding interactions, again, between the backbone, you can make a nice little very short turn. So say this would be part over here of a beta strand, have 1, 2 amino acids here, then start back in a beta strand going in the opposite direction. So very short turns that you can have between these other elements of secondary structure. Glycine also serves very well in these turn kind of structures because it only has a hydrogen as its side chain. And so that only hydrogen of a side chain gives you much more flexibility in terms of where you can put your individual protein. So secondary structures, how do those guys come together? Well, these are usually what people call motifs or structural motifs. I like the term supersecondary structure, so it's multiple secondary structures that are coming together. These are just a couple of those, so-called hairpin turn. So now you've got 2 beta strands and a beta turn, beta alpha beta, beta strand alpha helix, beta strand, helix loop helix, 2 alpha helices with a loop between them. The so-called Greek key motif, which is 4 beta strands, which are then hooked up to each other individually here again with small loops. Why do we care about motifs? Well, motifs very often have specific functions. And so let's look at a couple of these here. Probably the most famous of these is a so-called coiled coil motif, also known as the leucine zipper. And that's because you have 2 alpha helices that interact with each other by basically wrapping around each other. And you remember those side chains which are sticking out on either side. Well, if you've got side chains that stick out that can now interact with each other, and particularly in the case of side chains which are highly hydrophobic, and leucine being one of those, you can now have these leucines interacting with each other and literally interacting in kind of a case like a zipper. So you've got a leucine coming from this alpha helix, leucine from that alpha helix. 7 amino acids later, you have another leucine. 7 amino acids later, you have another leucine, etc. So these guys can hook up together and form very stable structures between these two. And a nice example of molecular complementarity, where you've got these 2 structures that come together. And we'll talk about leucine zippers as we move on to the rest of the course as well. You can also have these helix loop helix motifs, where you have here an alpha helix. This particular loop is what's called an EF hand. It's really good at binding to calcium. And lots of calcium binding proteins will have these helix loop helix motifs which come together. The last one, at least in this case that we'll talk about, are the so-called zinc finger motifs, where we have a zinc finger, and the structural biologists came up with this, but it's a helix and 2 beta strands that coordinate zinc, and very often this alpha helix interacts with DNA. So many, many DNA binding proteins have these alpha helices in zinc fingers. And back in the late dark ages, when I was an undergraduate student, my molecular biology class, they asked a question on an exam: can a zinc finger get caught in a leucine zipper? I'll let you think about that. So those are the motifs, and as I mentioned right before, usually those motifs by themselves don't actually have a structure. They've got to be attached to something else. And to actually have a structure, usually you have what's called a domain. And that domain are the collections of your secondary structures which in and of themselves have a stable 3-dimensional structure. And of course, your stable structure is probably going to lead to some kind of function. So a nice example here is of the influenza hemagglutinin. Have you had your flu shots? Flu shots? Yes, good. Bad year for them. Really good match with the flu vaccine. It's not too late to get them. Our speaker at the biology department in 2 weeks is going to talk about vaccines and how important it is to get them. But here, the hemagglutinin, which actually determines whether this is a H1 or an H3 or an H9. That H actually stands for this particular molecule right here. It's made up of 2 major domains. There's this distal domain, that's which is going to bind to the parts of your lungs or respiratory tract. Here is a proximal domain, which is important for holding the whole structure together. Two separate domains, each of them in and of themselves has a structure, has a particular function. This is a nice example of a protein which also has quaternary structure. So it's got the tertiary structure of these two domains, which come together as three separate polypeptides, trimeric form. So it's this multimolecular structure. You've got lots of different kinds of domains. Here's a nice domain which is almost all alpha helical. Here's a domain which is all beta strands, and often you have mixtures of these different things. So these are domains, and again, these have stable structures, very often have individual functions. Well, what about whole proteins or whole polypeptides? Very often, they're made up of multiple different domains, just like we saw with the influenza hemagglutinin. So you've got different pieces which get put together to give you an individual function, and it turns out that a lot of evolution seems to be involved with moving these domains around and giving them different functions to each other. So the nice example that we have here, I think this is from our textbook, is the so-called EGF protein, epidermal growth factor, not important for what it does here, but this is a very specific structure, and it can act just by itself, or it can be part of many different proteins. And so this particular one, how you make EGF in the first place, it's got multiple different copies of EGF in it, and then another domain which is attached to that protein. Some other proteins will have an EGF domain, and this is a binding domain. It's actually very similar to an immunoglobulin, that's why this got that structure. And tissue plasminogen activator also has one of these domains, but multiple different domains as well. So mixing and matching domains seems to be a really good way evolutionarily of generating new structures, and of course, generating new structures gives you new functions. We're getting there. Now, one example of a multi-domain protein is the RNA polymerase. This is the DNA-dependent RNA polymerase. Talked about before, this is the molecule which is involved in taking a DNA and making an RNA copy from it. Each of these different colored blobs here, which is represented here over here as the backbone of the individual amino acids, each of these blobs represents not just a domain, but an individual polypeptide. And so this is a multi-polypeptide protein complex. So this would be what kind of structure? Quaternary structure, exactly, coming together to make this really pretty amazing molecule. This was just published a couple of years ago, and again, Dr. Bartlett here in the biology department does research on this particular molecule. But that really pretty complicated molecule in and of itself is only part of the machine which is involved in actually making RNA from DNA. So if we look here, this is the cartoon of the RNA polymerase, and just to remind you, it's those 12 individual polypeptides that come together down here, together with a whole zoo of general transcription factors and the mediator complex, which all come together to start transcription. So this is beginning to try and give you an idea of the complexity of the macromolecular interactions that you have going on inside a cell, and that's what's keeping us in business for hopefully the next couple of generations here. So let's talk about how you go from that primary sequence of amino acids to those structures, and then eventually these really amazing molecular machines that you have down here at the bottom. Let's look at that process. All that protein folding is going from this stretch of linear amino acids hooked together with peptide bonds to a tertiary structure. This will be a domain, part of a protein, part of a whole macromolecular complex, which now has function. So how that happens, it's due to lots of weak bonds that are forming between different parts of this polypeptide chain here. We've got ionic bonds, we've got hydrogen bonds, we've got van der Waals interactions, etc. So it's all those weak interactions that are coming together to form this three-dimensional structure. It's another way of looking at that. Here again, we've got ribonuclease. It's a relatively small protein. You can take this protein from its final three-dimensional active structure, pull it apart completely, get it back to this linear form, and then put it back together, and it makes exactly the same structure with exactly the same function. And so these were the original experiments which said it's just that stretch of amino acids which is all the information you need to make an individual structure with a particular function. Now, one of the things that molecular biologists have been trying to do, basically since these experiments in the 1960s, is now figure out, if you start out with a stretch of amino acids, what kind of tertiary structure you're actually going to get. Because it'd be great if you could start with a stretch of amino acids and predict what the function of that is going to be. And we've been trying for almost 50 years, probably more than 50 years now, to do that, unsuccessfully. And the main reason for that seems to be that you think about 20 different amino acids all hooked together, one after each other. You have an almost infinite number of possibilities in terms of how all of these things could come together. And in many cases, they can, but they don't work terribly well. And this basically tells us evolution's a heck of a lot smarter than we are. Although admittedly, it's had a lot longer, 4 billion years rather than about 50 years. So that's basically what we're looking at here. So let's look at a couple of different problems with making those kinds of predictions. One of them is that you can have very different sequences relative to each other, the individual amino acids hooked up again from N to C terminus, that come together and give you almost identical structures. And so these are two examples here. This is the elastase enzyme and chymotrypsin. They are about 30% identical in terms of the individual amino acids that are hooked up next to each other. But they have almost identical structures here. A little hard to tell. You can squint a little bit. They look about the same. But most importantly, they do almost exactly the same thing. They're ACEs. Okay, so ACE, what does that probably mean? Enzyme of some kind. Turns out these are proteases. And they chop up proteins. It's supposed to be shown as the substrate down here, a little red piece. So 30% in terms of the number of identical amino acids between two different proteins is usually the rule of thumb in terms of saying if it's more than 30% identical, these two different proteins to each other, it's probably going to have the same structure and probably have pretty similar function. On the other hand, there are also a lot of examples of proteins that have less than 30% amino acid identity to each other, but again have almost identical structures, giving them almost identical functions. And if you think about it, again, from the evolutionary aspect of things, that's saying that there's evolutionary pressure to have a particular function, but there are a whole bunch of different ways to skin the cat. You can actually get that particular function, which means a particular structure, in a lot of different ways. You can use lots of different combinations of amino acids to actually give you that particular structure. So this example here is the so-called homeodomain structure. It's a helix turn helix structure. It's a DNA binding protein, really critical for development. And here the proteins from Saccharomyces cerevisiae, again, that small genome model organism that's been really well used in eukaryotic model systems, now at the top, and then Drosophila down at the bottom, the fruit fly. These guys are structurally almost identical with this little loop structure in between the two, and they're considerably less than 30% identical to each other. And that's just over this relatively short piece of sequence here. A lot of these sequences are in much bigger pieces. So how do you get these particular structures? Well, the first step, and I mentioned this before, is that we have one really big constraint in terms of protein structures. And that is that the peptide bond, even though it doesn't look double bonded the way we always draw it, It actually has a partial double bonded character to it. And that's because you have this what's called a resonance structure between this C double bond O and the CN here, which gives you a partial double bonded character in this peptide bond between this would be the C-terminal end of this amino acid and the N-terminal end of this amino acid, which you've hooked together to make your peptide. And what that means is this bond basically can't rotate. It's stuck in one plane. On the other hand, you have rotations here between the N and the C and the C and the C, and again, it's your carboxyls would be the next one to your carboxyl group, and then the next amino acid. These are also called phi and psi angles. The critical numbers are not important there, but this is basically the flexibility you have in terms of putting individual amino acids together. How does that happen? Most of the times it happens as the particular protein is being made. So here we've got our ribosome, which is mostly made of RNA. Yes, the ribosome is mostly made of RNA, making protein. And so here's that protein as it's being made. Proteins are made as primary sequence. So it's just one amino acid after the next amino acid, after the next amino acid, after the next amino acid. But that now has to somehow form its tertiary structure over here. And it's as that protein, I should say the peptide, is coming off of the ribosome that it has to form and make its actual final structure. And that structure is only one of, again, basically an astronomical number of potential structures that could be made. And so how that process takes place depends, of course, on those individual amino acids which are present there and how they're coming together relative to each other. Probably the most important aspect of those individual amino acids has to do with their hydrophilicity or hydropathicity. If you look at individual amino acids, arginines and lysines, those ones that have stable charges under physiological pH, those are extremely hydrophilic. What are they going to want to do? Go to the outside of your final structure. On the other hand, valine, leucine, and isoleucine down here are the hydrophobic ones. Those are the ones that are going to want to interact with each other, and then they're going to end up on the inside of your final structure. And so that gives you the oil drop model, which they mentioned in the textbook. Again, our hydrophobic amino acids here shown in blue, which are going to associate with each other because water is interacting with everything else. And then the hydrophilic amino acids, which are represented here in red, those are going to be the ones which are present on the outside of your molecule. What does that process look like? Again, here we have our primary sequence, an unfolded polypeptide chain, comes together, usually makes individual secondary structures. Those secondary structures associate with each other, and then finally form your folded stable structure, which again has now function. Turns out that there are lots of things that go wrong in terms of protein folding. Again, astronomical numbers of possibilities. The actual final protein that you get down here is everything. It's the N-terminus and the C-terminus, sometimes which are going to be really closely associated with each other. But those have been made in a linear fashion on the ribosome. So how do you get those things together? And in many cases, you have problems, and these things can aggregate, and as we'll talk about probably on Monday, a lot of diseases actually are involved in problems with protein folding. Probably the best known of those is Alzheimer's disease. Lots of neurodegenerative diseases have to do with inappropriate protein folding or the structures being inappropriate. So it's important that you get rid of those proteins that haven't folded properly, or you also have a way to help them be the right proteins that form. And so that process of helping are called the chaperones. And I love this term because I like to think of chaperones as stopping incorrect interactions. And as the father of two daughters, I think it's a really good idea, stopping inappropriate interactions. So basically the idea here is that you have now proteins which are helping to fold, but usually helping to fold by stopping those incorrect interactions from happening. So here there are really sort of two examples, and there are really two major classes of protein that do this. They're either those that are involved while that individual peptide is being made, and this would be the co-translational chaperonins. These are the HSP70s, the HSP90s that we'll look at in just a second. You also have the post-translational folding processes. These are the so-called HSP60s, the classic chaperonins, and we'll take a look at those in just a little bit more detail here right at the end. HSP70 and HSP90, these are the ones which are involved in co-translational folding. These have hydrophobic patches on those proteins. Those interact with the hydrophobic patches on your nascent peptide. With ATP hydrolysis leads to conformational changes in the protein. We'll look at that again in more detail later on. But that conformational change in the protein basically grabs the hydrophobic piece, lets it go, grabs it, lets it go, and makes sure that it interacts with the appropriate part of its own protein. So that's true for HSP70, HSP90, very similar in terms of how it functions. In this case, it's actually closing a structure in terms of hanging on to one particular part of a peptide, then opening it up again due to ATP hydrolysis. And then the last case, and this is what's happening in terms of already folded proteins, are the so-called HSP60s. I like to think of these as boxes of infinite dilution. That's what they call them. So basically what happens is you open up the lid on one of these, you throw an unfolded peptide into it, you shake it up, you've got a whole bunch of ATP hydrolysis that takes place, and then magic occurs and the protein, the folded protein, comes back out. And that's usually because what's happened, you remember all those hydrophobic things that are sticking out on the outside when you've got your wrong interactions, that, senior from high school, I don't want dating my freshman daughter. So that kind of interaction, you block that interaction and then surround my daughter with all of her freshman friends, and then she can go off and do her thing. So that's the idea as far as the chaperonins are concerned. Just wanted to mention in the last couple of minutes here that why all of these things are called HSPs. They come from heat shock protein, and it goes all the way back to those model organisms that we talked about last time and at the beginning of the lecture today. And that is, if you take an E. coli and you heat it up to hotter than it wants to be, it starts to make a whole bunch of these individual proteins, HSPs, heat shock proteins. And the theory, anyway, is that what happens when you raise the temperature, you start to have proteins come apart. You have those hydrophobic regions which are being exposed. Well, you want to block those. Well, how do you block them? You make more of these heat shock proteins. And so that's the idea in terms of why these should be heat shock proteins. Now we know that they're functioning also in the absence of heat shock. Turns out they're some of the most common proteins you have in a cell at any time. All of these folding proteins, because it's a really important process, and you don't want stuff to go wrong. So we'll stop there and talk a little bit about disulfides and peptidyl-proteolyl isomerases, peptidyl-prolyl cis-trans isomerases next time. Have a nice weekend.